So based on what we covered last time, let's just remember that molecules only absorb light at very specific wavelengths based on the structure of the molecule that's interacting with the visible part of the spectrum. It's for this reason that molecules have what we observed in lab a couple weeks ago, an absorption spectrum. And this spectrum, which plots the absorption as a function of wavelength on the x-axis and absorbance on the y-axis, is similar to what you collected in lab. This is different for each of the photosynthetic pigments that are shown here. Chlorophyll A has a slightly different absorption spectrum than chlorophyll B, and likewise with beta carotene. Those absorption spectra are what leads to the difference in colors of leaves, for example. When we enter into the fall, chlorophyll is being reabsorbed by the plant, broken down in the leaf and reabsorbed. That nitrogen source is being preserved for next season. And that leaves behind the carotenes and xanthophylls and some anthocyanins that appear red. And so that's why leaves change color. But for our purposes, thinking about photosynthesis, just know at this point that light energy is transduced into chemical energy starting with the absorption of light energy by these pigment molecules. And when we say absorption, what we mean is the energy that's in a photon of light is actually doing work on the pigment molecule itself. And the kind of work that it's doing is what we'll talk about next. What we call it when light is absorbed by a molecule is we say that that molecule enters into an excited state. So that is called excitation. The excitation energy is what is absorbed by the pigment molecule. Individual molecules that are in the leaf are becoming excited, or they're entering the excited state. And all that means really is that the electrons that are in those shared bonds for a molecule like chlorophyll, if we go back and take a look at that chlorophyll molecular structure back here, this chlorophyll molecule, its electrons are rearranging, and that's what we mean when we say excitation. The excited state is just the rearrangement of electrons as they orbit the carbons and nitrogens and that magnesium in the center of the chlorophyll molecule. When the excited state is formed, and remember that excited state represents the conversion or the transduction of light energy from the sun into now a chemical state. So it's now a potential form of energy. And it's changed state from electromagnetic to excited state of a molecule. What can happen is that that electron, or really the system of electrons that are shared in the complex structure of chlorophyll, can undergo an oxidation reaction. And remember, a molecule is oxidized when it loses an electron. And so photooxidation is light-driven departure of an electron. That electron is leaving the molecule when it absorbs a photon of light, it enters the excited state, and then that electron, it can depart that molecule that was excited, but only if an electron acceptor, something else, as we already learned and talked about multiple times, if something is being oxidized, then you know that something else has to be reduced. And in the case of the light reactions of photosynthesis, the system that allows for a chlorophyll molecule to lose an electron requires that another very similar molecule, very, very close in proximity, very nearby, a neighbor molecule, also has to be primed and prepared to receive that lost electron. So what we find is a donor electron, a reaction center chlorophyll as it's shown here. That reaction center chlorophyll can only lose an electron if there is an electron acceptor very near to it that's capable of receiving the electron. All of this is taking place not with free-floating chlorophyll molecules out in the leaf, But inside chloroplasts, and more specifically within the thylakoid membrane structure of the chloroplast, where chlorophyll molecules are, again, they're not free floating, but they're attached in very specific ways and orientations and at very specific sites on reaction center proteins that make up the photosynthetic electron transport system. It's through the arrangement of chlorophyll molecules attached to the atoms that make up proteins within the thylakoid membrane that allows for this very elaborate transfer of electrons when they enter the excited state by absorbing a photon as part of a chlorophyll molecule. That excited state that the chlorophyll molecule enters can donate an electron to a nearby electron acceptor. So what's shown here is the transfer of excited state energy from chlorophyll molecule to chlorophyll molecule without photooxidation occurring. And this is really a quantum physics phenomenon where the excited state itself can be transferred from molecule to molecule without moving electrons. It's only at a very specific set of chlorophyll molecules that are attached at a very specific place within the reaction center, photosynthetic reaction center, where the photooxidation reactions take place. All of the rest of the chlorophylls that are involved in absorbing light essentially transfer or funnel the excitation state itself to that reaction center chlorophyll. So we can picture that this excitation and electron transfer in this way. We can imagine a chlorophyll molecule being in a ground state. That's the kind of the basic lowest level energy state that the chlorophyll molecule is stable in. When it receives a photon of light energy, it enters the excited state, and that excited state chlorophyll molecule can then donate its electron to an electron acceptor. In this slide, the orange part toward the right. And when that electron acceptor receives an electron, it leaves the excited state chlorophyll molecule now oxidized. And in its oxidized form, it's lost an electron to the electron acceptor, so that acceptor is reduced. But the chlorophyll molecule is oxidized. And that oxidation state is really, really reactive. That chlorophyll molecule really now craves a replacement electron. And that electron, ultimately, if we trace it all the way back, that is the one that's going to be donated through the oxidation of water. So the donor molecule is shown here to the left of the center chlorophyll, and it's sort of squishy, maybe shaped like water. It doesn't directly come from water. It comes from an intermediate system that slowly removes, or piecewise removes electrons from multiple molecules of water all at one time, so that one O2 is produced. So in fact, there's a system or a series of reactions that occurs that's not shown here. But suffice it to say for now that the craving state, the oxidized state for the chlorophyll molecule that's lost an electron, it craves an electron so badly that it has enough energy to split water. And so that is a very powerful force in nature. It's the most oxidizing potential that we know of in nature. And part of that is because, water is really stable. It holds together really well. And so to break the covalent bonds between the hydrogens and the oxygen, that requires a great input of energy. In fact, that reaction, the reverse of it, when we take liquid oxygen and react it with hydrogen gas, that forms water, and that is energetic enough. That's how we lift rockets into Earth orbit. That's rocket fuel, taking liquid oxygen and hydrogen and reacting them together. So that gives you some proxy, some idea for how much energy is involved in this. All of the energy to do that comes first from oxidizing the chlorophyll molecule, and that oxidation is driven by light. So I've mentioned the idea of photosystems now a couple of times. Photosystems are the proteins found in the thylakoid membranes that attach chlorophyll molecules and really are the functional centers of the photosynthetic electron transport system. There are two photosystems in the thylakoid membrane, and they act like a series circuit. What I mean by that is that the products of one get passed along to and react in the second in the series. So they really chain together, pair, side by side. There are two photosystems, as I mentioned. They act in series, and these photosystems are embedded in the thylakoid membrane. So if we picture a leaf cell or a leaf cross-section as shown here in this slide, the leaf cross-section, if we zoomed in on one of the cells in the middle of the leaf, the so-called mesophyll cells, middle of the leaf cells, and then we zoomed in even further to the chloroplast. There are a few dozen chloroplasts in each mesophyll cell. If we zoom in even further to one of those, we see at the bottom of this slide the cutaway view of that chloroplast. The chloroplast, remember, it's the endosymbiotic organelle. It's got a double membrane system, so it's got an inner and an outer membrane. And remember, it has that specialized membrane system called the thylakoid membranes that are crisscrossing all throughout the inside of the chloroplast. The thylakoid membrane system is where we find the photosynthetic or the two protein-based photosystems. Those photosystems are holding on to chlorophyll molecules in, as I said, really particular, really specific orientations. Those chlorophyll molecules are membrane bound because of this long hydrocarbon tail. That makes them hydrophobic or nonpolar, makes them easily inserted into the membrane. So we find chlorophylls are membrane bound, and they're bound to proteins in the membrane. In particular, if we take a look at one of those thylakoid membrane packages, we see a photosystem shown here at the top of the thylakoid membrane system. And that, in this case, looks like it's a photosystem 2, probably, that's involved in the splitting apart or the oxidation of water. And so this forms part of what we call the light-dependent reactions. You remember, photosynthesis is not a single series or a single sequence or metabolic pathway, but it's a number of metabolic pathways that are interacting. One of them, we said, was the light-dependent reactions. The light-dependent reactions we can think of as light-driven redox reactions. Those are reduction-oxidation reactions. The energy to do the oxidation is coming from light. That's what we just talked about, the excitation state and the loss of an electron to an electron acceptor molecule. All of that is part of the first early steps of the light-dependent reactions. Water acts as our electron donor in these redox reactions, and it's oxidized to protons or hydrogen ions and oxygen. The electron flow, as electrons are removed from water, they pass through photosynthetic electron transport chain that's similar to that found in the mitochondria for the respiration electron transport chain. And that electron flow, again, does something very similar to what it does in the mitochondria. It forms a proton gradient. It's this proton gradient that forms, not in the mitochondria in this case, but inside the chloroplast, that forms this proton gradient that's used, once again, to make ATP, and ultimately those electrons that flow through the system and lead to the proton gradient being formed, those electrons get attached onto a temporary electron carrying molecule, NADP, to reduce it to NADPH. The electron or the proton gradient that's formed across the thylakoid membrane sees protons accumulate inside the lumen, the inner compartment that's bounded by the thylakoid membrane. If we go back a couple of slides, you can see here the lumen is the innermost compartment, and that's where our protons are going to accumulate. those protons that accumulate are used once again to drive the chemiosmotic or the photosynthetic phosphorylation of ADP to ATP. So those are the two major products of the light-dependent reactions, ATP and NADPH.